I'm Prishti. So moving on from the plasma membrane, we would move to the other part of the outer membrane, that is the cell wall. The cell wall basically is the outermost covering that has two primary functions. One is the protection and second is the providing of the shape to the cell. So what are the components which provide actually the shape as well as the protection are different, and that's how the various bacteria differ. So earlier we had studied about staining of bacterial cell. Cell wall plays a major crucial role in actual staining of the bacterial cell. So as we studied earlier, there are two types of bacteria based on the cell wall, that is the gram-positive bacteria and the gram-negative bacteria. So what the difference is between, apart from the differences, there are certain common moieties that are present. The cell wall basically consists of the peptidoglycan units or the murine, which provides the shape. So this peptidoglycan actually differs in these two kinds of bacteria. So what happens is gram-positive bacteria is the cell wall is primarily consisting of just the peptidoglycan units cross-linked between each other. So what does the peptidoglycan actually consist of is basically the N-acetyl glucosamines, that is the NAG, and N-acetyl uramic acid. So that is the NAG. Apart from that, there are 4 amino acids which are present inside the cell wall. So basic composition of the peptidoglycan remains generally common. What happens in gram-positive bacteria is that we have just the NAG and NAM residues which are cross-linked. In this case, the NAG and NAM residues are directly linked instead of cross-linked. Overall, apart from this, the gram-negative bacteria, the cell wall is actually a very complex one. Apart from peptidoglycan, they have LPS, that is the lipopolysaccharides. LPS basically consists of 3 things. One is the lipid A, the O polysaccharide, and the O antigen. So this actually forms the outer covering for these bacterial cells. So what happened is during the staining, when we had the crystal violet dye, so in case of gram-positive bacteria, the crystal violet actually permeates inside and actually binds to the NAG and NAM residues. So a permanent staining occurs. But in case of gram-negative bacteria, what happens is that the LPS layer actually blocks the crystal violet from entering into the cell membrane. So what happens is the staining is actually a temporary one. So as soon as we do a washing, the stain is permanently lost at this point. So that's why in gram-negative bacteria, we get a negative test, that is we get a pink colored colony that we studied earlier, and in the gram-positive bacteria, we have the violet color colonies. So apart from cell wall, we have various other structures that are present inside the bacterial cell. These structures include basically the internal membranous systems, there are various kinds of internal membranous systems that are present associated with the plasma membrane. One of them are mesosomes that I had already said about. Mesosomes actually perform the function that your mitochondria performs in the cell. So it is generally associated with the plasma membrane and performs the function associated with the plasma membrane. Plus, it has been found that mesosomes are generally prominent in case when the cell is dividing. So it is also thought to be associated with the nuclear division of the cell. apart from mesosome, there are various other structures which are associated with the plasma membrane and somehow kind of replacement to the organ keys that a eukaryotic cell would have, which are not present in the prokaryotic cell. But these structures, even mesosomes, may be present or may not be present, and these differ from the various kinds of bacteria that are there throughout. So apart from internal membranous systems, we also have the periplasmic space. This is the periplasmic space that we have. The width of the periplasmic space differs greatly between the gram-positive and the gram-negative bacteria. So, as we can clearly see, the periplasmic space is the space between the plasma membrane and the cell wall. So, what are the basic functions of periplasmic spaces? That firstly, it contains peptidoglycans, which would actually eventually be forming the cell wall. Therefore, since it has peptidoglycans, it would definitely have enzymes for peptidoglycan synthesis. Also, it has been found that the periplasmic space also contains certain enzymes for the ATC or respiratory system. So basically, in gram-negative bacteria, generally periplasmic space is very small because of the presence of the outer wall. So what happens is all these enzymes that are present in the periplasmic space, they do not exist in gram-negative bacteria. Also, the cell wall is also very thin in gram-negative bacteria as compared to the gram-positive bacteria. So these are the basic functions of a periplasmic space.